Welcome to Study Loop. Imatinib is a drug that is used to treat leukemia. It dissolves easily in water and has been found to build to very high concentrations in diseased cells. Recall that we discussed 4 ways materials move across membranes: simple diffusion, facilitated diffusion, active transport, endo, exocytosis. Refer to this yes/no decision tree darr for determining modes of transportation for chemicals. Given the information above, you should be able to eliminate some of those 4 methods as possibilities for imatinib. Which of the 4 transport methods can you eliminate in this case, and how do you know imatinib is not using those transport methods to get through cell membranes? In case it helps, there's a video about membrane transport gen you can review. Mostly complete, 10 PTS. Note that because this chemical moves to higher concentrations, goes against concentration gradient, that means it cannot be moving by any form of diffusion. So we can eliminate both of our forms of diffusion. The drug is hydrophilic, so that's yet another reason it cannot move by simple diffusion, which is reserved for hydrophobic chemicals. Let's examine each transport method's features in connection to imatinib in order to ascertain which of the 4 can be ignored for the drug based on the facts supplied. The passive transfer of molecules from a region of higher concentration to one of lower concentration without the need for energy is known as simple diffusion. It is usually saved for molecules that are tiny, hydrophobic, or nonpolar. It is possible to exclude simple diffusion because imatinib is hydrophilic and migrates to higher concentrations in sick cells, against the concentration gradient. The passive passage of molecules across a membrane is also a part of facilitated diffusion, although transport proteins are necessary for this process. Moving from high to low concentration, this mechanism nevertheless adheres to the concentration gradient. The elimination of facilitated diffusion is also possible because imatinib migrates to higher concentrations, against the concentration gradient. ATP is often needed for active transport, which moves molecules against their concentration gradient, from low to high concentration, by using energy. Imatinib can travel across cell membranes via active transport, which makes sense given that it builds up to large concentrations in sick cells. Cells use the processes of endocytosis and exocytosis to either ingest or eject big molecules or particles. Regardless of concentration gradients, these mechanisms are capable of transporting molecules. Endo, exocytosis may still be an option for imatinib transport in light of the evidence. In conclusion, based on the data presented, due to its hydrophilia and ability to move against concentration gradients, imatinib cannot be delivered by simple diffusion. For the same reason, it is not possible to transfer imatinib through facilitated diffusion. Simple diffusion and enhanced diffusion are therefore the 2 transport mechanisms that can be disregarded. The only viable routes for imatinib transfer across cell membranes are endo, exocytosis, and active transport.